This is a Holy Cross mock for component 2, and this is mock number 1 out of 4 possible mocks. And we're on to question 5. Things you don't want to hear in an exam. Coat colour in mice is determined by at least 3 genes. Things you do not want to hear. Right, and we've got one of them is dominant B for black and recessive allele B for brown. And we've got another gene where we've got dominant A for something agouti and little a for non-agouti. And agouti is just the colour changes along the hair. Right. So we've got a description of those, and we know about dominant and recessive. So all good so far. In a dihybrid cross, a black agouti mouse was bred with a brown non-agouti mouse. Okay. So we must be able to identify the parental phenotypes because they've asked us to do that, and genotypes. And well, the phenotypes that they've just given us those. So are they giving us a mark for that? They are. Oh dear. So it's a nice start. They're giving us a mark for just repeating something in the question. So parental phenotypes. Black, agouti, brown, non-agouti. Yep, they're giving us a mark for that, just repeating that from the question. Okay, some of the offspring were brown and some were non-agouti, right. So this is a classic sort of question where it tells us what the dominant is. The dominant, the capital letters there, and we're crossing them together and we're getting, some of the offspring brown, some are non-agouti. This must be, it says recessive, and the genotype is recessive and the phenotype is showing the recessive trait, so we know what this one is. If it's brown, it must be little b, little b. So little b, little b. Non-agouti is also recessive, that is little a, little a. So we know that one. But what's this one? If this was homozygous for the dominant, so if it was big B's, then all of the offspring would be black, because there'd always be a big B there, not big B. And the same here, if it was monohybrid for big A, all the offspring would have an A in, and so all of them would be agouti. So this one must be heterozygous. So it must be big B because it's black, but must also have the recessive allele. Big A because it's agouti, but also the recessive allele. We're making sure also that we're not breaking any golden rules. Each of a kind of normal cell will have 2 copies of the gene, so 2 alleles present. So we'll always have 2 B's and 2 A's. 2 B's, 2 A's. That will always be the case unless it's gametes. And the next bit is, well, let's choose the gametes. And we know about random assortment. And so this gamete is going to have half the genetic material. So it's going to have 1 copy of B and 1 copy of A. This is a frequent error to put, 2 B's down here. So you have 1 copy of B, 1 copy of A. So that's one possibility. But it could have been that copy of that B and that little a. possibly. Or it could have been the little b and the big A, or it could have been the little b and the little a. So that parent has 4 possible gametes. Here we're tempted to do the same thing. It could be a little b and the little a, or a little b and the little a, or a little b and little a, or a little b little. That sounds ridiculous, doesn't it? There's only one possible gamete there. And so there's a nice helpful space here for us to draw a Punnett square. And so I'm just going to put these gametes down the side. Being careful to copy them out as I have put them. And across the top we've only got that one. And if I was to draw a box around those, a mini Punnett, and we can do the adding up exercise. So big B, little b, big A, little a, big B, little b, little a, little a, little b, big A, little a, and this last one, little b, little a, little a. And so our F1 and genotypes are these, and our phenotypes, well, anything with a big B is going to be black. Two little b's, that's going to be brown. And anything with a big A is going to be a agouti. Two little a's, it's going to be non-agouti. Non-agouti. So I could write, I'd have to write these out here, or if I left it like that, I'm sure they'd give us the mark, but however, I'll write them out. Big B, and I'm going to separate these just to make it really clear. That's black, agouti, black, non-agouti, little b, big A, little a, brown, agouti, and the last one, all homozygous recessive, brown, non-agouti. So I've made it really clear which phenotype belongs with which genotype. In a litter of 8 offspring, how many would you expect to have whatever is brown agouti? So they are looking at this one, brown agouti. Well, it's got a 1 in 4. So 1 in 4, how many out of 8? So it's a 2 in 8. So 2 out of 8. So 1 in 4, and then you just times it up for how many you've got. And now, here we go, we are looking at a third gene. A third gene, and hopefully you've not given up by this point. It controls the production of the pigment melanin. So you've got melanin, it gives you your colour. Mice which are homozygous, so the homozygous have the recessive allele little m, and it tells you what they look like, and those are albinos. So the genotype of the albino, or genotype for the homozygous, that's pretty straightforward. Little m, little m. Be careful with your m's, make sure they don't look like big N's. And by what process might a dominant allele spontaneously change into its certain blah blah? Mutation. So if an allele's changing or base is changing, that's a mutation. So not too bad so far, this. And the general term applied to agents that increase the frequency of such changes. So what makes more mutations? And it says a general term, so not any examples on X-rays or radiation or 20 Benson edges. It wants a general term, so these are mutagens. So mutation generators, that's how I think of that. In large populations and in the absence of selection, the proportion of dominant to recessive alleles will remain constant from generation to generation. That's fairly straightforward. And some definitions are. Definition for, I love it when they just ask us for definitions. Gene pool, all of the alleles present in a population. So all, that's the key thing, all of the alleles present in a population. You can be a little bit flexible with the word alleles. You might have genes, or population you might have in a particular place. But that's our normal definition for a mark. Genetic drift. This is the kind of random changes in the allele frequency. So random changes in the allele frequency, particularly with small populations. Lots of things happening due to chance. So a couple of just definitions there. Explain how owls might reduce the frequency of the albino. So owls, famous for seeing in the dark, they will presumably eat the white mice because they can see them, but we need to try and use our genetic words and from variation and evolution. So owls select against the albino allele. So how might they reduce the frequency of the allele? Because they select against the albino owl. Owls can see the white mice in the dark, just in case. Not quite sure where that mark's coming from. And these mice will be able to reproduce and pass that allele on. And another version of the same thing, how hawks, which hunt in daylight, might reduce the frequency of the allele B, which is where is that? That's our black. So hawks presumably can see the black mice in the day. So hawks select against B in the day. Prevent it being passed on, that sort of argument. And the last couple of marks explain why over many generations the allele B might completely disappear, but allele M will always be present. Well, the clue there in the capitals, might disappear, small lowercase characters might always be present. Well, the answer is really because in our dominant, if something happens against our dominant, well, the dominant's always going to be presented in our phenotype, and so if something happens to it, there's a possibility that our dominant might disappear, whereas our recessive allele might be hidden in the heterozygotes. And so it's very difficult to get rid of it because it always will be lurking around, hidden behind a dominant allele. So we just need to express that. So recessive alleles can be hidden in heterozygotes. Dominant alleles always visible and can be selected. Again, it's not much space there for two separate points. And that's 70 marks.